next speaker will be Laura Nicolás-Saenz from the Universidad Carlos III. And he will tell us about a new registration technique. Okay, so whenever you want, Laura, if you are ready.
Hello. Can you hear me?
Yes, we can.
Great. Okay. So, hi, my name is Laura Nicolás, and I'm here to present a very recent paper that we've published, which is called Nonlinear Image Registration and Pixel Classification for Pipeline for the Study of Tumor Heterogeneity Maps. This is the main idea index that we're going to go through. So first of all, what was our motivation for this work? Well, in cancer research, as most of you will know, pathologists study biopsies taken from the tumor and stained with different proteins and antigens. These antigens are used to mark biological functions as well as the areas where the tumor is most active. So for example, in here, the brown means inflammation in this particular biopsy. With this information, they obtain a score depending on the amount of antigen, which in this case would be the brown, present in the tumoral area, which in this case would be the red area. For example, in here, let's say we'd get a 70% score for inflammation. With the different antigen distributions, we can give important information on the heterogeneity of the tumor. We can get a 70% inflammation, 40% proliferation, 50% for angiogenesis, and this leads to a personalized prognosis. So this can be used for the creation of personalized therapies, and this would be our goal, to go from trial and error with chemotherapy and horrible treatments that are very hurtful and painful to the patients, to personalized treatment that we would be more sure that it would work for each individual patient. So for this, we need new tools that are robust, fast, and automatic. This, in general, means computational pathology. But the problem with this is that it usually needs training. So the idea is clear, but there are several challenges that right now need to be overcome. First of all is the staining protocol. There are no two biopsies that are stained the same. It varies within different laboratories, within different countries. Even within the same laboratory, one stain can be incredibly different from the very next one, because it's a very difficult thing to do well. Also, the visualization, the images are huge. They take a lot of computational resources, and they are very difficult to work with. And the tumor heterogeneity, each tumor is completely different, and it's very difficult to decide which therapy would be best for each one. So because it is very difficult to integrate and analyze all of this, we need help. So our goal for this is to create a series of probability maps that can integrate all of this information and explain the behavior of tumors and their biological functions and how these proteins are distributed, in order to maybe select better therapies or help with research. And all of this should be automatic and non-supervised, so that we do not add any toll to the pathologist's already difficult work. So for this, we use images that were publicly available from the Automatic Non-Rigid Histological Image Registration Challenge from last year. We used two sets of images, the gastric adenocarcinoma and lung lobes. And we had 56 whole slide images, which 8 different markers in total, and a total of 38 landmark files. These images presented with the usual rips, folds, losses of tissue, and other artifacts. Here we have some examples of the gastric set, in which we can see the missing tissue. In here, the rips, the folds that were present, and in the lung lobe set, the same. The images can never be perfect, because taking these slices is very difficult. Also, we can see that they were huge. They were between 2K and 5K pixels each, which is a lot for a computer to manage. So our general pipeline was to, from these images, including the landmark files, proceed with an automatic registration pipeline, then a segmentation pipeline, and with those two create antigen masks that could be developed into a heat map, a probability map of how these proteins are distributed within a biopsy. So for the image registration, we followed a general pipeline of 3 steps. First of all, we followed an alignment step that was basically displacement and controlled rotation to solve this rotation and displacements without any constraints. So the easiest part of registration, really. Then a global motion model to describe the overall motion of the moving image with some deformation limitations. For this, we used an affine transformation and a Demons transformation. And finally, a local motion model to take into account all the local internal deformations that constitutes the actual morphology of the biopsy, with huge deformation limitations so as not to completely change the internal morphology. So with this, we would introduce a rough binary mask of each slide, and we would get a registered image. And this registered image would go through a pixel classification pipeline, which uses the red-blue color joined histogram. This is a double histogram of the red and blue channels of each image. And here we see an example with a tile of an already registered image. And we use the main diagonal to separate this histogram into the purple population and the brown population. And with this, we can create the masks for the purple and brown regions. Now, we obtained quite good results. We are very proud of them. We used the relative target registration error to evaluate the registration performance, because this was the proposed metric in the challenge from which we took the images. And we effectively reduced the median error in each step. We got a final median error of around 0.007, which means a better performance than a lot of state-of-the-art methods, which are ANS, RVSS, B-on-Warp-D, and NIF-Direct, and a very similar performance to the other 2 state-of-the-art methods, Elastic and DROP. In here, we can see the different steps of the registration and how the images and the landmarks were affected by them. And we can see that after the local transformation, after the last step, the images are almost perfectly aligned, and a lot of the landmarks are perfectly overlapping. And here we can see the importance of this local transformation, because after the global transformation, in which we do not take into account the internal morphology, yes, the images are generally aligned, but the internal structures are a bit of a mess. And after the local transformation, the images are well aligned, and the internal structures are also well aligned, are also corresponding. For the pixel classification, we compared our algorithm with the very famous WECA's trainable segmentation, in which you train a classifier to discern between the different pixel populations. Our algorithm did not need training, and overall we achieved basically the same results. So we managed to get a good registration step, a very good segmentation step. The only thing left was to create the heat map. And here we go. So with each of the different stains, we could create a map that explained how the proteins were localized and were interacting with each other in the whole biopsy, right? Not in just one slice. So we could integrate maybe 5 different images of the stains into just one map. And in this case, for example, we can get a lot of key information from just one image. So for example, in here, we can say that the endothelial cells, which are the CD31, are located around the lesion, that the inside is dominated by pneumocytes, that divided nuclei are located within the lesion, either by themselves or with pro-SPC, and that Clara cells are only found lining the small airways. And here we have another example with a gastric dataset, in which all the same, we integrated 4 different images, 4 different stains into just one map. And with just one image, one visualization, we can get a lot of key information, for example, about how the T cells are distributed and how they are the majority of the stain, and how the other immune cells, the lipid antigen presenting molecules, histocytes, and cytotoxic T cells are mostly present around the edges. So we can get a lot of information from just one image instead of trying to compare 4 different images, going through all of them, going back, going forth, and being generally quite confused. So this could be used in many different ways. It can be used just as it is as an aid in assessment of biopsies and for scoring tumors. It can be used for pattern prediction if we go into research of different treatments for specific cancer, for personalized medicine. And it can be used as an automatic, non-supervised, computer-generated input for training deep convolutional neural networks, which are old-fashioned today. So thank you very much for listening to me. We hope we enjoyed it. If you are interested, in here you have the paper we published this past August. It's in the Entropy magazine. And well, I hope you enjoyed it and that you generally liked it. Thank you very much.
So thank you very much, Laura. Can you hear me?
Yes.
Okay. So we didn't receive not yet questions in the chat, but I have a couple of them. Okay, why do you choose the red and the blue channel and you reject the information about the green channel? I'm not an expert in that, so it's just a very basic.
Right. Well, the green channel usually mostly contains information about light, but not necessarily about the stains. And we figured by looking at a lot of tiles that the red-blue histogram had this very conserved shape that could be divided by the diagonal. And this was conserved over all the different immune stains that we tried. So we decided that we did not need the green channel at all. And we continued with this.
Okay, and there are some, for instance, in your slide number 32, you can see some stripe noise. So there are some vertical stripes there, and are they related with the algorithms that you apply for these transformations, local transformations, and so?
Right. No, in this particular case, it was all a matter of how the biopsies were taken. And then the stains had that particular stripy nature.
So it's a kind of a mark of the knife used for the, the biopsy.
Most likely, yes.
Okay. Yeah, we are receiving now some questions. So let me pass you some of them.
Okay.
Okay. So Mafalda Sousa says that she needs some clarification. So why do you need registration steps to get the correct heat map?
Oh, right. Because am I sharing my screen?
Yes.
Okay. So because this is generally, this is how we receive our images. So the images are rotated, they are displaced. There is a lot of artifacts due to acquisition. So there is stretching in the tissue, there's folding in the tissue. So if you were just to try and align them without any registration step, you wouldn't get anything. You would just get a mess.
Okay. Second question. From Keith Jalink, sorry. Can you say something about how calculation intensive this is compared to other methods?
Sorry, how what?
How the computational cost compare with other methods, or the time that you need compared with other methods?
For the registration?
Yeah.
Okay, so for the registration, the first 2 steps are pretty straightforward and they hardly need any time at all. So maybe in 1 minute it's fine, it's done. The last step, local transformation, which requires free-form B-splines deformation, is very computational costly. That's true. So it needs quite a bit more time. It obviously depends on the computer capacity. So if you need to do it in a very normal computer, it will take you probably quite a few hours. If you do it in a very good computer, it will take you about 20 minutes.
Yeah,
and
but
it is something
I think that the main thing is to if you make a fair comparison with I mean, with the same computer, with other methods and your method, can you say something about the advantages in the registration, in the time that you need compared with the others in the same conditions, of course?
Right. In the same conditions, our method would be the longest, probably. It will take the most time. But we are working on that. We currently have quite a few good ideas on how to reduce this. And I mean, it is right now, it is true that it is not super efficient for the results. But we think that with the new implementations that we are currently going about, it will be quite efficient and quite worthwhile.
Okay. So let me check for some other questions. Okay, yeah, Diego Mejías asked about how do you deal with inhomogeneity in the staining of the different parts?
Right. So great question. A very difficult thing to do generally in this type of things. Thing is that surprisingly enough, our pixel classification method works no matter how bad the staining is, no matter how different the staining is. It just does. It's wonderful. So we don't really have to do anything, I'm afraid. There's no pre-processing other than segmenting the background, really. The rest of it, it works.
Perfect.
It's very robust.
So great.
Yeah.
Okay, so does this method, Veronica Labrador asked, does this method works for different color stainings? I mean, you are working with a given set of dyes, but does it work with many different stainings, or did you try?
Yeah, so far we've tried with these 8 different ones for these 2 data sets. We've tried with a few other stains. I believe it was 5 other stains that were from the Nonlinear challenge as well. And then the year before we tried with, was it 8 different stains from a hospital here in Madrid. So we've covered quite a few different laboratories and different stainings, yeah.
Okay. So there's more technical questions. Just one more because we are reaching the end of the session. Iranzu Conde asked if you say something briefly about Weka trainable segmentation.
Oh yes. Well, I have an extra slide here for that. Weka trainable segmentation is a marvelous method that was created by Ignacio Arganda. And it is really, right now, I'd say the state of the art in pixel classification. So the general idea is that you just input tiles, a tile for training. You do manual labeling of whichever number of classes you have. So you label, let's say about 15 labels per class is usually enough. So you label that for a tile, and then you just introduce the rest of the tiles, and Weka does the job, and it creates, yeah, a semi-supervised segmentation of the images. You also have a different choice of classifiers, so you can use random forest, perceptron classifier. You have your choice. You can play quite a bit with it. It's quite fun, really.
Okay, so there are some extra questions. We don't have time for that, but please, I will recommend you to check the chat. Okay. When you go to the hop-in session, check the chat, and if you want, you can ask directly in the chat the questions that are the remaining ones.
Okay, great.
Okay, thank you very much. Okay, I think that for now, this session is closed, and now we have a break, and we have the possibility of meeting the speaker, Manuel Martinez, at the proper session room. So if you have any questions, please go there, and we will start again by 11 with a sponsor, a new sponsor debate. So thank you very much.